Welcome to this lesson on dihybrid crosses. A dihybrid cross is a fundamental concept in genetics that helps us understand how traits are passed from parents to offspring. Unlike a monohybrid cross, which examines just one trait, a dihybrid cross allows us to examine two different traits simultaneously. This powerful tool helps us predict the probability of offspring inheriting specific combinations of characteristics from their parents. A dihybrid cross is defined as a genetic cross between two parents that differ in two distinct traits. The purpose of studying dihybrid crosses is to understand how two separate genes are inherited independently from one another. This concept was first studied by Gregor Mendel, the father of genetics, who discovered that different traits are passed down to offspring following predictable patterns. When we perform a dihybrid cross, we can observe how these two traits combine in various ways in the next generation. Let's explore the classic example using pea plants, which Gregor Mendel famously studied. In this dihybrid cross, we examine two traits together. The first trait is seed shape, which can be either round or wrinkled. The second trait is seed color, which can be either yellow or green. By crossing plants that differ in both of these characteristics, we can observe how nature combines these traits in the offspring. This example perfectly demonstrates the principles of dihybrid inheritance. To work with dihybrid crosses, we need to understand genetic notation. For seed shape, we use the letter R. A capital R represents the round allele, which is the dominant form. A lowercase r represents the wrinkled allele, which is recessive. For seed color, we use the letter Y. A capital Y represents the yellow allele, which is dominant. A lowercase y represents the green allele, which is recessive. Remember, dominant alleles mask the expression of recessive alleles when both are present together in an organism. Now let's set up our dihybrid cross by identifying the parental generation, abbreviated as P. Parent 1 has the genotype RRYY, meaning it is homozygous dominant for both traits. This parent produces round, yellow seeds. Parent 2 has the genotype RRYY, meaning it is homozygous recessive for both traits. This parent produces wrinkled, green seeds. When we cross these two parents together, we can predict what their offspring will look like by analyzing how these alleles combine. When we cross the parental generation, all of the offspring in the F1 generation, which stands for first filial generation, will have the genotype RRYY. This means they are heterozygous for both traits, carrying one dominant and one recessive allele for each gene. Because R and Y are dominant, all F1 offspring will display round, yellow seeds. Even though they carry the recessive alleles for wrinkled and green, these traits remain hidden. This uniformity in the F1 generation is an important observation in dihybrid crosses. To understand the F2 generation, we need to examine gamete formation from the F1 individuals. When an RRYY individual produces gametes through meiosis, the alleles separate and can combine in different ways. Each F1 parent can produce 4 different types of gametes. These are RY, 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 and RY. Each gamete receives one allele for seed shape and one allele for seed color. This independent assortment of alleles is key to understanding the variety we see in the next generation. To visualize all possible offspring combinations, we use a 4x4 Punnett square. Along the top, we place the 4 possible gametes from one parent, RY, 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 and RY. Along the side, we place the same 4 gametes from the other parent. By filling in each box where gametes meet, we can determine all 16 possible genotype combinations in the F2 generation. This systematic approach helps us calculate the probability of each genotype appearing in the offspring. When we analyze the phenotypes, or observable traits, in the F2 generation, a distinct pattern emerges. Out of 16 possible offspring, 9 will have round, yellow seeds, 3 will have round, green seeds, another 3 will have wrinkled, yellow seeds, and 1 will have wrinkled, green seeds. This gives us the famous 9:3:3:1 phenotypic ratio. This ratio is characteristic of dihybrid crosses and demonstrates how traits assort independently during inheritance. Let's review the key takeaways from dihybrid crosses. First, Dihybrid crosses allow us to track the inheritance of two traits simultaneously, giving us insight into how multiple characteristics are passed from parents to offspring. Second, the classic phenotypic ratio is 9 to 3 to 3 to 1 in the F2 generation. Third, each parent produces 4 possible gamete combinations due to independent assortment. Finally, dihybrid crosses beautifully demonstrate Mendel's law of independent assortment, showing that genes for different traits are inherited independently of one another. Understanding dihybrid crosses is essential for predicting inheritance patterns in genetics.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.